All right, so hello, I'm Lindi Wahl. I'm a mathematician. Oh, you can't hear. Oh, on. Hello. Should I move it up? Hello? Is it better? Speak loud? Okay. So at least I sit in a mathematics department. So this talk will be a little bit more mathematical. What I'm going to do is a kind of historical overview of models of adaptation. So it'll be a little bit like a review paper. And you know how review papers have those little boxes sometimes? In fact, one that was the reading had lots of little boxes. And we're going to do a little mathematical box. I'm going to take a piece out of the middle and work on the board. And then we'll come back to the review with some more contemporary work. So modeling adaptation. Oh, I think I can use this. Beautiful. So adaptation is the evolutionary process by which a population becomes better able to live, that is, survive and reproduce in its habitat. And it's a little bit confusing because selection is not exactly the same as adaptation, right? So selection means that alleles that are more fit increase in frequency in the population, but that doesn't imply that the population is getting better able to survive and reproduce. Can you think of an example where selection does not produce adaptation? It's hard to come up with one. Thinking. Yeah, that's a great example. Sexual selection, right? So the peacock's tail is selective forces have put the peacock's tail in place, and yet we don't really think that it's adaptive. It doesn't help the peacock population survive better in its environment. Right? Yeah, great example. So fundamental to our ideas of adaptation is this idea of a fitness landscape, which was introduced by Wright. Oops, that's not the pointer. By Wright, long ago. And this picture is a little bit confusing because these are allelomorphs. You probably can't see that. But let me show you what I would draw instead. You might think of points where this is genotype big A, big A, big A, little a. Over here we might have little a, little a. And then vertically, I'm going to draw their fitness. So this, for instance, might be the most fit. And these guys are somewhat intermediate. Maybe this is the least fit allele. So wherever we are in this landscape, there's a fitness associated with the genotype. That's the fitness landscape, right? And if we had many, many genotypes, we could kind of approximate, at least in our minds, as a kind of continuous surface where, again, the vertical axis is fitness. So down here, there's a genotype that's not very fit, and here's the best possible genotype in this population. Or perhaps we might think about this being phenotype space instead, and we'll talk more about that. And then adaptation is the process by which the population might start here and then climb up towards the peak as it adapts, right? Becomes more fit. So historically, here's where the history begins. Darwin himself, and right through to the 1930s, sorry, gotta get used to the buttons, believed that mutations were very small and adaptation would have to proceed through a series of very tiny changes. There's a nice quote from Darwin: "Natural selection can never take a leap, but must advance by the shortest and slowest steps." And yet it seemed to be a bit of a paradox because Mendelian genetics, we have discrete genes or discrete heritable units, and how can that be reconciled with these teeny tiny steps that adaptation is supposed to work through? And these ideas were reconciled to some extent by Fisher in the 1930s. So he argued that we could approximate adaptation by a series of tiny steps. Even if each step is discrete, if there are many, many steps and each step is really, really small, it's almost as if things are continuous. And that, of course, laid the foundation for quantitative genetics, which you've heard a bit about, I assume. And you heard a little bit this morning already. And in quantitative genetics, phenotypes, as I guess you now know, are summarized by their mean and their variance across the population. more standing genetic variation. And this is a picture of quantitative genetics, adaptation in quantitative genetics. This little cartoon looks like this. So here we have time, and here's some value of the trait, the length of the peacock's tail. Oh no, that's not adaptation, let's not use that. The tail feather length of some bird that needs a long tail feather to fly. And this, you can imagine these poking out of the page, right? That's the distribution of types in the population. So most birds have feathers of this length, but then in the next generation, if there's some selection for a longer tail, the population moves there and there and there. And Fisher also introduced an idea which is going to be the subject of the box, the blackboard part of the lecture, Fisher's geometric model of adaptation. And it's been really influential, and I think it's very interesting. And there's also some really interesting recent work, so that's why we're going to talk about it a bit more. But I'll just introduce it now, because this is where it comes in the historical perspective. So in Fisher's geometric model, we think about axes in space as being phenotypes, right? So the realized value of the trait that we could measure, and the trait is continuous. So instead of axes like this, we'd have the length of the tail feather, and I don't know, the beak length. And then fitness is some surface here. And somewhere there's an optimum beak length, then an optimum tail feather length. The best birds are there, the most fit. And mutations might take the population or an individual from the population, well, from these 2 points, which would have that fitness. A little mutation might take the offspring to there, which would then could then have higher fitness. And there's a picture from the reading. This, I can only draw 2D, not even very well. That's a 3D picture of Fisher's geometric model. So now we have 3 traits, and each population lies at some point in 3 space. And populations that are on each concentric sphere have equal fitness, and the highest fitness is in the center. So this is an adaptive walk. The population, some random mutation occurred. It was very beneficial. It fixed, and the population moves to here. Then another random mutation occurs. It's beneficial. It fixes. The population moves there, and it climbs. This is hill climbing, right? And space is continuous here. Yeah. So no. Yeah, no. Space is continuous despite the fact that we've drawn these are kind of like the isoclines in 3D. And as we'll see in more detail in a minute, Fisher showed that the smallest mutations have the highest probability of being beneficial. And he used this result to sort of strengthen his argument that mutations during adaptation, the ones that contribute to adaptation, are very small, and tiny steps are made. I love this picture because then this is when Mutukumura said, there's something wrong with that argument. And you can kind of see him going, So Mutukumura pointed out that the mutations that contribute to adaptation, not only do they need to be beneficial, but they have to survive drift. And drift, you've just heard, right? It's this random change in allele frequency. But when a new mutation occurs, drift is really, really important to the life of that new mutation, which is something I'll talk about in a couple days in more detail. But the allele frequency over time, these pictures that Brian showed, when a brand new novel mutation occurs, it starts way down here. And then all of this bouncing very frequently bounces it down to 0 and it disappears. Right? Most trajectories are not successful. And very rarely, even if the mutation is quite beneficial, very rarely does it survive drift. I don't know if you've talked about that yet. So Kumura argued that mutations have to survive drift, and they're more likely to survive the more beneficial they are. And again, I'll show you some more pictures of that. later, but he argued then that mutations of intermediate size might contribute to adaptation most, not the smallest ones. However, that argument was sort of set aside because intellectual effort at the time was focused on the neutral theory, which was nicely introduced already today. So as you heard, we were just saying over the break that we should have coordinated our lectures maybe a little bit more. But I think it's good, actually, that you hear it again, you hear it in a slightly different way. It'll slowly start to sink in, right? So many single nucleotide changes are silent, and therefore their effects are probably neutral or nearly neutral. And most substitutions that are observed in molecular evolution are nearly neutral and have just been fixed, we assume, by drift. And here's a nice picture. This is the landscape or the distribution, frequency distribution of mutational classes. That's not really so clear, but these are the available mutations. This is their selective cost. It's a cost if it's below 0 or benefit if it's above 0. So their cost or benefit, and this is the distribution that we expect. So there are many nearly neutral mutations. There are many deleterious mutations. I would even add probably a spike down here of many lethal mutations. And advantageous mutations is kind of, you can't even see them there. They're very rare. But then if we look at the frequency distribution of mutations that actually fix, the substitutions that we observe, the deleterious mutations very rarely fix. They're purged from the population by selection. The neutral fixation, or the neutral mutations, have a pretty good chance of fixing by drift, particularly in small populations. And there are some advantageous fixations, more than there were advantageous mutations over here, right? Because if you're very beneficial, you're much more likely to survive drift. And so through the 1960s and 70s, a lot of people did some really important work, which I'm not going to tell you about. But it was mostly focused on purifying selection, the elimination of the deleterious alleles. And the prevailing thought was that positive selection or adaptation was very rare. We wouldn't observe it, and we wouldn't see it in the genetic information that we had. An exception to that was John Maynard Smith, about whom we heard some lovely anecdotes last night. Although not too many people were working on adaptation, Maynard Smith was an exception at this time, and he argued that evolution occurs in discrete sequence space. Ultimately, genes are discrete. The nucleotide base pairs have to change in a discrete way, and only genetically close sequences are accessible by mutation. And so adaptation must be a walk that's a sequence of discrete steps, even if they're small. So he returned to this idea that we need to have discrete steps in adaptation, that really things aren't continuous. And he made this quite famous. You'll see this quite a few places in his Nature 1970 paper, this discrete sequence changes that will take you from word to gene. And we're constrained by the fact that all the organisms in between word and gene have to be viable, so they all need to be real words themselves in English. It's just a little analogy for how discrete steps have to proceed in adaptation through viable intermediates. But according to Orr in the reading material for this lecture, this work was almost entirely ignored for 2 decades. So then what happened after 2 decades was that quantitative trait loci, QTL, I don't know if you've seen those yet. Yeah, I can't really tell you much about them because I'm a mathematician who doesn't understand them, but molecular markers are used somehow to find the genetic basis underlying phenotypic differences. So it involves some ANOVA, like analyzing variances. And many studies began to find QTL, so single loci that had very large phenotypic effects if they were mutated. And sometimes a quite small number, a handful of QTL, each of which had substantial effect, would sometimes underlie species differences. So this pointed to not only were steps discrete in adaptation, but sometimes the steps were quite big. And this was kind of new, right, at the time. It was a big insight that came QTL. And then in the 1990s, we've heard a little bit already about experimental evolution of microorganisms. Populations followed in the lab for thousands of generations, and adaptation is observed. This is, I believe, a yeast population. It's a single SNP, basically, an SNP. And when I say large effect, I mean these guys can grow 10 times faster. They change one base pair, and suddenly they grow 10 times faster. Really large effects are possible. So evidence from QTL and from experimental evolution has sort of renewed interest in the modeling community to model adaptation, either as a walk through phenotype or genotype space, or something which I call, it's more like an adaptive gallop. You know in a gallop, that some feet are up while the other feet are still coming down, and it's all happening at once. And this is, I think, characterizes some of the more recent work on adaptation, especially in large experimental evolution populations, where the adaptations are all sort of happening one on top of each other. And, well, Kavita and Michael know a lot more about that than I do, actually. So the questions that are of interest there, if there's an adaptive walk, how many steps happen? How big is each step on average? Can we make any kind of general conclusions about how adaptive walks happen? How fast do they happen? How fast is the gallop, if it's happening? In what parameter regimes do we get a walk or a gallop? And what's the distribution of selective effects of the mutations that fix? Okay, so now we will move to the blackboard. Can I get the projector off? See me being much more relaxed because this is my typical modus operandi. Once I have the chalk in my hand, I'm way more comfortable. So we're going to start with Fisher's geometric model, as promised. And I know like every single person that's talked up here has asked you, but just so, because I was sitting at the front, I couldn't really see. So how many people do have, say, undergraduate level training in math, physics, engineering, these kind of hard sciences? Oh yeah, that's good. That's actually like half or more than half. Good, so this will give you something to chew on. Because this, and if you don't have training and some of this stuff, just let it just wash over you. Enjoy the mathematics for its elegance, except for Deepa. She told me that she sees no beauty in mathematics. Okay, so we already said that the axes are going to be continuous trait values, so quantitative traits. And I drew a picture already, and I'm going to draw it down here. I'm going to flatten that picture out a little bit. So we'll have, trait A, wingspan, and trait B, let's do the tail feather again. Tail feather length. And each individual is somewhere on these axes. But actually, Fisher's model is not about individuals, it's about populations. So we think about the population being at one point in these axes. So say here, the current wild type population. But there's some room for adaptation. They're not at the best possible place. And the optimal place to be would be over here. And then fitness decreases. Fitness is coming out of the board, right? The peak is over here, coming down. You see that? There's fitness. And Fisher argued that mutations would be like little vectors of a random length and a random orientation. So randomly chosen angle, randomly chosen length from some distribution, and a mutation might take trait values over here, in which case it would have lower fitness than the wild type, and it would be purged. But a rare beneficial mutation might happen that took trait values here, and it would have higher fitness than the wild type, and it might fix, and then the population would move to here. And the random walk in 2 space is a little easier to see. Might move towards the origin over time. And what do I want to write down for you? Now imagine that the length of the mutation vector is really small. So I'm going to zoom in. This guy's sitting on a fitness isocline here. If I zoom in there, and I add a really small mutation, in a randomly chosen angle, what's the probability?
So actually, you assume that there's just one sort of wild type. Is that a reasonable assumption, or do you want to put some sort of variation to it?
Yeah, that's a good question. In the classic Fisher's geometric model, you just assume that the population is uniform, genetically uniform. There's no standing variation, but that's an excellent point. Yeah, so you could more realistically want a whole little distribution here. Yeah. So what's the probability that this mutation is beneficial? Oh, yeah? Yeah, well, that's another good question. So the question was, what is optimal fitness? What does it even mean? So there's an assumption in this model. If I tip it on its side here and we looked at the fitness landscape, there's an assumption that fitness versus trait has some shape like that, and that the best possible fitness is the peak. That's what the optimal fitness. We don't. Yeah, so yeah, the question was, how do we know that there is a best possible fitness? We don't really. We just assume, and we don't need to really know what it is for the model, but we just assume that it's reasonable that there might be one, which is a questionable assumption. Absolutely. In fact, there's a lot of argument about that. Yeah? Yeah. Yeah, if you like, I don't have that, we can call it max. To me, it's the same if it's optimal or maximum, but either way. Yeah. Yeah, beautiful insight. So the insight was, it's very hard to see from this sketch that most of the mutations would be neutral. And in fact, you're right. In Fisher's geometric model, most of the mutations are not neutral. Right? Absolutely. So you don't get the distribution. Actually, let's put an origin over here. This is your current wild type fitness, and this is the selective effect. Or, well, let's just say fitness. If I put fitness on this axis, I've been showing you pictures like this, right? Yeah. Oh, sorry. Well, you know what this is. Anyway, you don't get this bulk of mutations that are neutral out of this model. Yeah. So these are all wonderful points. Okay, so now you have to answer my question. I've answered all your questions. So what's the probability that this mutation is beneficial? Roughly. Yeah, and let's continue making them really small. In the limit, what am I going to get? 0.5, yeah, 0.5 exactly. So we have, if the length, let's call this length R, it's kind of the size of the mutation. How big is its effect? And if R is small, we approach 50% beneficial. Okay, what if R is really big? Like suppose I start here and my mutation is the length of the blackboard. Then what's the probability that it's beneficial? Zero. Thank you. Yeah, it's zero, right? Because you're always going to go right past the point, and you're going to go further away. So in the other limit, you get zero. R big, we're going to go to zero. And if you really want to know, Fisher worked out quite beautifully that the probability that it's beneficial is 1 minus the integral of a standard normal u du. So what's that? 1 minus the area under a bell curve. Up to point R. What's that? There's your bell curve. Here's the area up to R. So when we got to 0, we should be at 0.5. And if we went a little bit further, we'd be a little bit higher than 0.5, right? Approaching 1. And 1 minus that looks like this. Not to insult your intelligence. 1, 2, 3. Where actually these lengths are measured in some kind of normalized, we have to do some standard normalizations to make it all make sense. But the big insight is that indeed 50% of the beneficial, or 50% of the very small mutations are beneficial, and this is going to drop off pretty rapidly for large effect mutations. Yeah. So the question, or the point was, it assumes that mutations occur at any angle with equal probability. Yep. Okay, then Kimura. I promised to talk to you about Kimura's insight. There's something wrong with this argument. Over lunch? So the question was, can I do a little sketch of this proof? And I'm like backpedaling fast and saying, maybe over lunch I'll remember how that argument goes. Okay, sorry. So Kimura said, you might have some distribution of available mutations, as we saw previously, but the probability that they survive drift is very tiny, where this is fitness, or let's call it selective effect, the effect of the mutation on fitness. So there's 0. We should cross 1 on n here, 1 on 2n, and then go up like this. So as I said several times in words, the probability of surviving drift for a novel mutation is much higher if it has a big effect. And we need to take this into account when we look at which mutations are actually going to contribute to adaptation. So we actually need the product of that with the what kind of unknown distribution of mutational effects, but let's say it looks like this. So this is available mutations. And if we take the product of those two to see what substitutions we'll actually see, we might expect a lump kind of over in this direction. This is very hand-wavy, right? But this is the essence of what Kimura saw. And he said mutations of intermediate effect were more likely to contribute. And that's very hand-wavy as well. But the idea is that it's not the smallest possible mutations that are contributing. To adaptation. Okay, so that's the historical bit on Fisher's geometric model. And now I'm going to tell you about some more recent work on Fisher's geometric model, which I think is really interesting and a little tricky to understand. But I'll give you the basics, and if it captures your interest, I can easily give you the papers. Because I think there are a lot of fascinating open questions in this particular area, and it requires people with quite a bit of analytical training. So maybe I'll give somebody some ideas. So more recent work. From the classic model? So before we get, there's more recent work that calculates more based on the classic model, but classically it's really just this quantity, the probability that mutations are beneficial, of which size. So the guy to look up is Guillaume Martin, who's done a lot of this work at Montpellier. And now I'm really going to get mathematical here. So the basic idea is that I drew 2 trait axes, but we could have many, many traits. So let's have n traits. N continuous traits. And the population is characterized by a vector then, an n vector z, which is z1, z2, zn. And it's what they call a genetically coded phenotype, but we'll just call it phenotype. All right, so there's a point on each axis. And then a big assumption here is that fitness is going to be a multivariate Gaussian. So a bell curve, but in many dimensions. Which I will attempt to sketch. So there are axes, but actually I need the origin to be in the middle. There's the origin. And then fitness is going to be vertical. So somewhere above this point is the peak in the fitness. And we've got a Gaussian coming around. That's pretty good. I guess I should put the fitness axis up here. So this height is the fitness of the origin, and without loss of generality, as we like to say in math, we're just going to say that the optimum occurs at 0. So we're just going to shift the axes. So the optimum occurs there. And the current wild type is at some other point. This is on the flat plane at some point Z0 and has fitness W of Z0. Yeah, you with me? Yeah. Or those of you that want to be with me at least are with me. So what else do I need to tell you? Oh, so that multivariate Gaussian, it will look like this. the fitness of any phenotype will be e to the -1/2. We'll do Z transpose, is it S? S is what he uses. Z. And S is the selection matrix or selection covariance matrix. Okay, so that'll make sense to a lot of you, and if not, doesn't matter, just picture that. And we're going to just shift things so that the peak is at 0. Actually, 0 vector. Okay, keep going with the math. Population starts at Z0. We will define an S0 just because it's going to be useful as the relative fitness compared to the optimum 1 plus S0. So this is just defining a selective advantage. Have you seen? Sorry, I'm assuming that you've seen how S is typically defined. Do we know that? Yeah. Okay. So this is this selective difference between where the population currently is and where it could be if it were in the best possible spot. All right. Okay, and then there's another big assumption. We're going to assume that Z0 is close in some sense that I'm not going to define, but is defined very clearly in the papers to the optimum, or maximum, or the best phenotype. So the population is fairly well adapted. Okay. And what this does is allow us to assume that S is small. The relevant S's that are going to happen, if this were an adaptive walk, are all going to be fairly small. This is a really key assumption that Martin makes. And the reason he makes it is to do this trick. I think it's a trick. So therefore, S0 is small. And so S0 we can write as a log of 1 plus S0. Right, Taylor expansion. And you think, why would you do the Taylor expansion this way? You usually use Taylor expansions to make complicated things look like simple things, but we're actually doing the reverse. Exactly, yeah, because then we're going to stick it in here, and the exponential is going to go away. So this is actually, I think, Martin's big insight that allows all the math to be doable from there on. So yeah, you saw right through it. Yeah, that's a good question. So now we have S0 is log of W, Z0, W0. Without loss of generality, again, we'll just define this reference fitness to be 1, which will make this go away. And as you saw coming, we'll get S0 is, what, -1/2. Z0, S, Z0, a quadratic form, right? And the question was, why don't you just start with a quadratic fitness to begin with? And it's a good question. Yeah. Well, there's, I find this work really interesting, but it is not, well, I wouldn't say it's not elegant, but it's, he doesn't shy away from convoluting things in ways that make them more difficult, not less difficult to understand. Okay, so now we're ready for a mutation. We're just set up the whole thing where the population has started. Now a mutation happens. Well, I guess it happens down here. Right, changing. One individual has different trait values. And a mutation is going to take Z0, maybe changes with a little vector of changes, DZ. And what do we want to know? We want to know the S, the selective effect of the mutant. And this one we're going to do relative to wild type. Question? Okay. So we need S is log of the fitness of the mutant relative to the fitness of the wild type, log of Z plus DZ, Z0 plus DZ. The wild type is Z0. You see we're going to get some nice subtraction here. Maybe I'll leave that part up. Works out to be something like this. Probably more detail than you need. Another quadratic form. That piece minus the denominator. And this bit we already defined. By definition, that was what we called S0. Okay, we're almost there. For those of you that are just letting it wash over, we get the selective effect of the mutant is some constant S0 minus a distribution, gamma. And then I have, like in my notes here, it turns out lots of math, many appendices later. So if you go through the appendices, you can get finally that gamma is a sum of, I'm sorry, maybe I shouldn't even write this on the board because it won't mean too much. Maybe it will to some of you that have the right training. And the xi, these are kind of like the zi, but they're transformed. Again, we have to do some normalizations, just like Fisher did. Basically, we're diagonalizing. Okay, and speaking of, lambda I are the eigenvalues. Oh, I missed something, sorry. The eigenvalues of S product with M. And sorry, this is in my notes and I just skipped it. What is M? And M is the mutation, it describes the mutations, which mutations are possible. So this is a covariance matrix for the distribution of the DZ. I just threw the DZ on the board without telling you where they came from. So they're randomly chosen. So here's trait 1 and trait 2. And instead of just having in the very first bit that I told you about Fisher's geometric model, we had mutations of a given length distributed at equal angles. And then Martin has relaxed that so that you can have a distribution which defines where these mutations might come from. So you could get mutations that tend to increase both the length of the tail feathers and the wingspan, for example, right? And rarely you can increase one or the other. So you can have some covariance described by this matrix M. Yeah. So the question was, is it similar to steepest descent or ascent, the line of steepest ascent? No. So this is all about a distribution of possibilities. So they're not assuming that the move rule where you go next is the best possible place. But I will come back to that. Some other models do make that assumption. Yeah. So the question was, does M need to have special properties for this to work? And yes, SM has to have rank, has to have full rank, I believe. So there are some special properties, but none of which I can easily give good intuition for. So sorry. What I just drew? Well, for the later conclusions that I'm going to show you, yes, you need to, they assume that it's Gaussian, and then they can get even further. Yeah? So it's a multivariate Gaussian again. It's a good question. Okay. So this then, have you guys got statistics training? Hands up, I know some statistics. Okay, okay, yeah. So this, I'll just breeze over this. This turns out to be a sum of chi-squared distributions, which is sort of simple and known about. Okay, so the nice thing is that we get this distribution that essentially the S values will be distributed in this way that we know something about, a sum of chi-squareds. But they're independent chi-squareds, and so it's given some data, you could never really back out anything from that. But given some further assumptions, so with some further assumptions, so one, I already assumed that this S0 was quite small, and I'm actually going to reiterate that. We now need the xi to be small, which in the transformed coordinates, in sort of, this is kind of accessible mutation space. You're very close to the optimum. So we can think, if we start in words, you start close enough to optimum or maximum, best phenotype, that most of, how did I say, the distance to the peak is less than the size of most mutations. So you're really starting up here. And two, if all of these lambda I eigenvalues, are all the same, so what does that really mean? That means something mathematically, but what does it mean physically? It's sort of hand-wavy that the mutations in every trait are kind of equally accessible and equally selectively important. Okay, so these are really big assumptions, right, which you heard lots about yesterday, thinking about your assumptions. So mutations, kind of equally accessible and equally selected. Oh, I should say mutations for each trait. Okay, yeah. No, because initially it was I'm going to come back to that. It's a really good question. So this assumption that you're very close to the peak was not part of Fisher's initial model. That's right. Yeah. So these assumptions are just being made to get a solution. And you get a nice solution, but it's a lot of assumptions. Yeah, I'm going to come right back to that. This particular assumption is a little bit, this assumption is a little bit heinous. If we want to talk about adaptation, in fact, it takes adaptation away. Yeah, well, let me come back. Let me come back to this. I think this is a really important point. It's great that I think you're like getting your teeth into it. It's really good. Mm-hmm. Mm-hmm. Actually, the reverse happens. When you start so the question was, when you're that close, do you always just climb because things are fairly flat? There's not much curvature anymore. But actually, when you're so close that the distance to the peak is less than the size of your mutations, you're mostly going past. You're stepping over and you're going down all the time. Yeah, yeah. Or down in your own direction, but okay, so great. Exactly. This is really good. So now the comment was, then most of the mutations will be deleterious. Yes, exactly. So let me show you. Such a good crowd. You guys want to take first year calculus from me? I'd trade you in a second for my first year calculus course. So under these assumptions, where was I? We get S looks like this for those of you that really want the math. There's only one eigenvalue. Some number of traits. The dxi's, remember, are the changes that we made to each trait with the mutation, only in our transformed diagonalized coordinates. And this is, S is distributed as a gamma distribution. It's actually, well, it's got a negative sign in front. So we'll call it a negative gamma distribution, which is a really lovely, very well-known distribution, right? And as you were all intuiting, the distribution of S then, well, maybe I should just put it here. Yeah. Mm-hmm. S naught minus gamma. So, yeah, then we did, well, the diagonalization, well, we assumed we're so close that S naught is effectively 0. And the gamma was the sum of chi-squareds. And then when you have, if this is the way selection works, and this is the way your mutations tend to happen, I should have drawn this before when I did the diagonalization, where this is Z1 and Z2, the actual traits. In transformed coordinates, you get X1 and X2, and you get just one Gaussian here. Yeah. And then under the additional assumption that the lambdas are all the same. Well, I'm not doing this very well. This is now circular. That was elliptical. Lambda 1 equals lambda 2 equals lambda gives us this. And then that gives us the negative gamma distribution. And the gamma looks like this. There are no advantageous mutations. So all of our assumptions have brought us down to basically moving so close to the peak that we're effectively on the peak and we're only looking at deleterious mutations. Okay, yeah? Yeah, so the question, yeah, so there are further papers that move away from the peak and give you a distribution over here and talk about that. Yeah. So what I want to tell you. Okay, so what does this all mean? How much time do we have? We get to here. So if you start very close to the optimum, your mutations are Gaussian, your fitness is Gaussian, then you can get to this gamma distribution. And the gamma distribution has this nice feature. Not only is it a gamma distribution, but we know its parameters. And the cool part, or I lost it. So we get a gamma distribution, and it has scale, parameter lambda, and shape parameter n over 2. Okay, and this is actually really exciting. I know you failed to capture my excitement for this. This is really exciting because, well, lambda's like the eigenvalues of the SM matrix. We don't really care, right? But do you remember what n was? Number of traits. The number of traits, right? And so what Martin and his colleagues then did was to fit data from mutations, which, as you know, are by and large deleterious. So if you measure a whole bunch of mutants, you get a lot of deleterious effects. And they fit it to a gamma distribution and then pull out a number of traits. And they call this like the effective number of traits seen by selection. It's as if selection is happening in a 3-dimensional universe or a 10-dimensional universe. And for different organisms, it can be different. Right? So when they did that, what did they get? I wrote them all down. This should really be a slide. I apologize. But for vesicular some virus, they got 1. E. coli, they got 0.21. Seems a little suspicious to me. Yeast, that's the estimated number of traits. Yeah. Yeah, exactly. It's the number of traits somehow seen by selection. It's kind of like an effective population size, an effective number of traits. So the adaptive process, or in this case, the process of deleterious mutations, is happening as if there was only 1 trait. Yeast, they got something like 0.9 to 1.8, depending on whose data they used. Drosophila, you'll be happy to hear, are more complicated. They have 2 traits. And C. elegans was the most complicated organism that they had data for, 2.6. Okay, so there are so many assumptions taking us to this point that I think it's really questionable what the effective number of traits mean. Is this valid? Which assumptions were broken? But I think there are a ton of open questions in this work. But what I like about it is that its theory about these basic processes, that they fit to data and at least came up with a prediction. And there's still a ton more work. And as mentioned, I was just going to finish the little box about Fisher's geometric model. There this is like a 2006 paper, the one that I mostly talked to you about. And there are further papers that move away a little bit and actually get a distribution if there were some advantageous mutations, so we're a little bit further from the peak when we start. And they also look at modularity. They thought maybe we don't get so many traits because really organisms are in modules, and mutations only affect one of those modules sort of at a time, so a sort of subset of traits, and different mutations affect a different subset of traits. And this reasoning was invalid because of that, so they redid it with modules, but they didn't find much difference. So I think that's all I wanted to say. Do you have more questions about this stuff? Yeah. Mm-hmm.
So, I mean, are they able to sort of predict like the time scale of adaptation that
Yeah, not this stuff, but that will come up in my next set of slides. There's some work that does more adaptive. That would be what I think of as an adaptive walk. How many steps? Yeah. Okay, yeah.
Sorry, I'm not sure I got this, but what happens to
Neutral mutations. Yeah, so they go to zero if you're sitting right. The question is, what happened to neutral mutations? If you assume that there's a peak and the wild type is sitting on the peak, there are no neutral mutations, right? So the fact that we know there are many neutral mutations. Well, if there's, yeah, okay. Yeah, if they're completely silent. Yeah, that's right. That's a good point. If they're completely silent, they don't affect the phenotype, right? So they are not mutations. You just sit there. But there are, you could also say that there are, depending on the population size, right, we consider some of these nearly neutral. So for different population sizes, different windows around zero are considered nearly neutral.
So this model we started off with an assumption of small mutations and a continuous trait, which means that there are essentially infinite number of traits which are affecting the infinite number of genes affecting the trait.
Mm-hmm.
But then we are coming up with these discrete numbers. So isn't that contradictory?
No, there are infinite alleles, or you could say loci, affecting the trait, right? But there's one trait or two traits or three traits. Each of them can vary continuously and are underlain by a number of genetic effects. Yes. Yeah, so that would be in the M matrix. Yeah. The question was, how does the model capture, if I understand correctly, capture a one particular locus changing several traits? It's in the M matrix. And mm-hmm. Yeah, so the question was about multiple fitness peaks. In a fitness landscape, you might expect there are several peaks, local maxima and maybe a global maximum. You could say that this just isn't valid for those situations, or you can just imagine we're sitting near the top of a local peak, and mutations aren't going to take us further away, so we'll only consider our worldview from the point of view of the population. We're on one peak. Whether there are other peaks out there, we actually don't know or don't care. Okay, so now I'll go back to just a few more slides. I think I've just got 15 or 20 minutes. So I can do this. Pick this up. No? Okay, so that's the end of the little box, and now we're just going to pick up kind of the historical story again. And the more current thinking, well, not that this stuff isn't current, involves sequence space. And we think about the dynamics in different regimes in which adaptation has very different behavior. And these three regimes, I think of three anyway, are characterized by how many mutations we expect, the population size times the mutation rate. How frequent are mutations? And in fact, I'm just going to call this the beneficial mutation rate because I'm going to talk about adaptation again. So we want to know how frequent are beneficial mutations. If they're not very frequent, we have the strong selection, weak mutation regime. I'll tell you more in a minute. And then if they're increasingly frequent, we move to clonal interference and then the multiple mutation regime. So I'm going to tell you about those three. And here's a picture lifted from Barrick and Letsky's review. It's a nice review in Nature Genetics, recent, on experimental evolution. So here's what they call periodic selection. So this is the strong selection, weak mutation regime. So here we have genotype frequency versus time. And you can see that at some time, some beneficial purple phenotype arose, or genotype, I guess, and fixed in the population. And then at later time, this is a selective sweep. And then after purple has fixed, a blue guy arose. And you see successive sweeps, one after another. And the key idea here is that beneficial mutations are rare enough compared to the time that it takes to fix, that they only happen one by one. So we wait a long time, then a mutation occurs, it sweeps to fixation, then we wait again. And after some time, maybe we're lucky we get another beneficial mutation, it sweeps to fixation, and so on. This is an adaptive walk model. In clonal interference, actually as I think defined in the original papers, clonal interference, I'm sorry, my world is very haploid. So this is in asexuals without recombination. Clonal interference happens in asexual populations, and we have the original, there's a beneficial mutation happening again, but before it's, well, actually this one fixes, and then the yellow guy happens, but before the yellow guy can fix, another beneficial mutation, blue, happens. And because there's no recombination, yellow and blue cannot mix. They just have to fight it out. And in, I think, classically, if I can call it classic, since it's from 1998, clonal interference, the paper would assume that either blue or yellow has to win before we get the next mutation happening. And they didn't do a picture of multiple mutations, but actually multiple mutations is shown here in the sense that we can have, before the blue or the yellow fixing, new mutations are happening on top of these backgrounds, and then new mutations are happening on top of those backgrounds, all simultaneously. This is the gallop where the feet haven't reached the ground yet before the other feet are coming up. So strong selection, weak mutation. Like I said, beneficial mutations fix one by one. So the time to fix is shorter than the waiting time for another mutation to happen. Happens when this product is small. And John Gillespie did some really important work on this in the 1980s. And he argued, his big insight really was that whatever the underlying distribution of selective effects, beneficial mutations are samples from the extreme right end tail of that distribution. So let me sketch a bit. Okay. Oh, actually, you know, I think I actually found a picture of this. I may not need to sketch. Aha, okay. But let me back up and say, so tails of distributions, if you do have some statistics training, have some very common properties. Even when the rest of the distribution looks completely different, often the tails look the same. That's called extreme value theory. And Gillespie took advantage of these properties to derive some results. And here's the picture. So this is a distribution of selective effects. It might look like this, and the population might adapt. But as several of you argued, we expect that organisms in their environment are fairly well adapted. So they're probably somewhere up here. And if we zoom in, we have this tail of the distribution. And the tails of a lot of distributions are exponential, a lot of well-behaved distributions. So using these general results of an exponential distribution, Gillespie was able to make some really robust conclusions about adaptive walks. So he showed that they're probably quite short. So if you start as a fairly well-adapted population, after 2, 3, 4, a handful of steps, a handful of fixation events, you're probably as close to the peak as you're ever going to get. Because as you get closer, the number or the fraction of mutations that are still beneficial is declining. So we might expect bursts of substitutions to be a signature of adaptation. That is, the environment changes, the population takes 2 or 5 steps to climb back up to the peak. Then it sits there and only has deleterious mutations to purge for a while. But then if the environment changes again, we might see another little burst. And this is another area of fairly active research with recent extensions looking at things like the distribution of available selective effects and which ones would actually get substituted, how big are the steps, and current thinking is that fairly large jumps could be expected to happen. So half of the total fitness gain should come from step 1 on average. That's a pretty big statement, pretty big prediction to make. Following from that, at least chronologically, were NK models. This is a completely different class of adaptive model. I'm not going to give you details about the NK models, but they do work in sequence space and explore landscapes that are smooth or rugged. So many peaks or one peak in the adaptive landscape. And I think you probably know about smooth and rugged peaks, smooth and rugged landscapes, maybe. So a smooth landscape has one peak, which can be smoothly approached in all directions. And at a sequence level, it means that if nucleotide X at position Y is good, it's always good. It doesn't matter what the rest of the sequence is. So there's an optimum base pair at each position. All right? Yeah, so any other change doesn't affect the fact that you want to have, or say, let's say amino acid even, you want to have alanine here. It doesn't matter what else you have in your genome, you want to have alanine there. That's the best thing to have at that position. Yeah, it's a really strong assumption. Right? So we pretty much know that adaptive landscapes are not completely smooth. Right? Rugged is the other end of the spectrum, where changing one nucleotide in the sequence has a completely unpredictable effect. So if you change one position, the fitness is completely unrelated to what the fitness was before you made that change. It's just another random draw from your fitness distribution. So that's also, we know that's not really true, right? Exactly. Reality is somewhere in between. Yeah, you're getting worried, like there are no. The nice thing, so I think the power of the NK model is that you can move smoothly between these extremes. And it was the first model that allowed us to move smoothly between these two extremes, right? Because if Fisher's model completely smooth, and you can just have random draws from your fitness distribution for every sequence, it's also fairly easy to envision, have a completely rugged landscape. And the NK models are a kind of compromise where we can explore this space in between that otherwise, before this, wasn't really explorable mathematically. So there's the picture. Smooth, rugged. Here's a more interesting picture, I think, because this is fairly recent data from Aspergillus. And Franke et al. Plotted, so this is a bit hard to get through, but I think we can understand it. So here's fitness relative to some optimum, maximum, wild type. And this is just the number of mutations away in sequence space. This is like Hamming distance, right, away from this guy. And the lines show one-step mutational neighbors. So this genotype is one mutation different from this guy, and so are all of these. And then these guys each have a set of one mutational neighbors. Right? And some of these will be in common, so if you went here and then there, you could also get there by taking this step first and then that step. Yeah. Okay, and so what are the colors? The colors show peaks. So the colors are genotypes for which all their one-step mutational neighbors, at least the ones that they were able to measure in this lab, were less fit. So this guy, he's a peak, well, he's a global peak. All of his neighbors are less fit than him. But way over here, or here, this is not a very fit genotype, and yet all of its one-step mutational neighbors are less fit than it. So that's a local peak. And you could certainly imagine if you started here and tried to adapt, you could get caught on one of these local peaks. Right? You make a few beneficial mutations, you end up here, and now if you can only access one-step mutations, you're sunk. Okay, the block model is similar to the NK model. This is a lot of work. This work was done by Orr as well, but it was originally developed by Nelson and Perelson. Perelson. Mackin and Perelson. Thank you, Mackin and Perelson. And it is like the NK model, but it's a bit more intuitive. It's easier to understand and easier to explain to biologists. So now we think of the genome having independent blocks, which are like modules, tasks that this genome has to perform. And the fitness depends on the sequence in each block. But within the block, all the other, well, there's epistasis. So the fitness, let me draw it. We'll have 3 blocks. The fitness of this block depends on what sequence is there. And F2 and F3, and we're going to add them up. You can, depending on what question you want to answer, you might want to look at a range of numbers of blocks. Okay, and as I said, Orr did some work as well on, on these adaptive walks in the sweep dynamics. He considered a single step. Well, no, he considered not just a single step. Sorry, now I'm getting tired. But he looked at the whole adaptive walk and looked at the substitutions that are made through the walk, and in particular their size. And he was able to show under a set of assumptions, as always, that mutations are exponentially distributed, the ones that get substituted. So there are a few large effect mutations and very many small effect mutations are on average predicted to be substituted during an adaptive walk. And the expected size of these mutations should fall off geometrically. He also showed under, again, a set of assumptions. So if you expect the size of the first mutation to be S1, then the second step would be, say, 90% of S1 on average, and the third step would be 90% of 90% of S1. Yeah. So that means that your first step in adaptation will be the biggest, and then the sizes will drop off. And that's actually been seen in many experimental evolution studies now. And the move rule, we had some questions before, right, about the move rule. How do you decide where the next step is? So the model, basically, you have to say in your model how to choose which mutation will be the next one that sweeps through. And the NK models and the block models typically chose some random mutation. So kind of like Fisher's geometric model. We just take some random mutation and we move there if it's beneficial. But more realistically, Gillespie introduced the fact that we should also think about the fact that the mutations need to survive drift. And they do so roughly proportional to their S value. And so we should say that the next step depends not only, or isn't random, but would be more likely to be a larger mutation, a more beneficial S. Yeah. So clonal interference, as I said, now we get clonal interference is when new beneficial mutations occur while the first beneficial mutation is en route to fixation, and then they have to battle it out. This only is relevant to asexual species. And you expect that the largest beneficial mutation will outcompete the others and will fix. But the implication of this, the interesting implication is that there's some kind of speed limit on adaptation. So in the absence of clonal interference, you might expect if you increase the population size, you're accessing more and more beneficial mutations. And so you can get really big beneficial mutations, and they'll just fix, and adaptation will get faster and faster. And it should just keep growing. But there's a speed limit imposed by clonal interference, and this, I think, was a really nice insight. So that adaptation, the speed will saturate, by which I mean as we increase the population size, instead of seeing the speed of adaptation, which is how rapidly the fitness changes, instead of seeing that, we see this. And also, the distribution of fitness effects of mutations is changed by clonal interference. So this is a nice figure from another review. No, this is not a review. Nice paper. So here's some distribution of fitness. We don't really know what it looks like. Fitness effects of mutations. But we really only care about the tail. So here's the selective advantage of beneficial mutations, and it's probably exponential, as we argued. Although, again, there's a lot of work and controversy about that. And then if we apply the fact that more advantageous mutations are more likely to fix, and clonal interference, we get this distribution of mutations that actually fix. And then as you increase the population size, in general, this is important to have in your minds, things just get more deterministic, right? And so you access more beneficial mutations, and you can boost up. Clonal interference will make, give another reason why more beneficial mutations are more likely to fix and more likely to outcompete their neighbors. So this will shift up. Right. So then various people thought, but if beneficial mutations can happen in the wild type while the first beneficial mutation is en route to fixation, so this is this picture. You've seen it before, but I'll put it up here again. So while this is en route to fixation but hasn't got there yet, a new mutation happened. A different color arose here, and maybe this guy's actually better, and he rises to fixation. That's clonal interference. This S is bigger than this S. But if it could have happened here, the new mutation could also have happened here. In fact, if you look at when I drew it, it's more likely to happen in the wild type, but over here, you're quite likely to get a beneficial mutation here on top of this first one. And so the beneficial mutations are accruing and all happening at once, and this is the multiple mutation regime. And as I said, there are people in the room, names you will recognize, that know more about this and have written papers about this, and more about it than I do. But I will try to talk to them about their own work without being too nervous. So the first models to predict adaptation rates in this regime assume that all beneficial mutations have the same S value, and this was really just to make things solvable. It really makes the math much easier to handle. And it removes this complication surrounding competing mutations of different size and who's going to win. And there's more recent work that allows for a distribution of mutations with different S values. And I just wanted to say, especially to this crowd, that much of this work is being contributed by researchers that have really strong physical and mathematical training. And there's still beautiful open questions here. The math is really terrifically interesting. The physical intuitions are terrifically interesting. And there's many opportunities to test these results by computer simulation. And ideally, we're getting predictions out of these models that are testable by experiments, right, in microbial populations. So this is another area which I won't tell you too much about, except to encourage you to look into it if you're looking for some good problems to solve. Yeah. And talk to people over lunch. Okay, I think that's it. More questions? You were great with your questions. Really fantastic. Okay, let's have lunch.